everyone, my name is Luisa Gómez. I'm super excited to take part in the BAT Paper Advent Calendar of 2020. I think it is a great initiative to share with other BAT researchers what other people are doing in this field, and I really think we should continue doing this during the following years. I can imagine that you already heard a lot of stories in the past days about cool papers that people have shared with you. And today I want to talk to you about a unique and extraordinary species, which is the common vampire bat, Desmodus rotundus. The paper I'm going to tell you about is a follow-up of a series of lab studies that have been conducted on sickness behavior of vampire bats. It was published on behavioral ecology, and it was led by Simon Ripperger, Sebastian Stockmeyer, and Jerry Carter. The name of the paper is Tracking Sickness Effects on Social Encounters via Continuous Proximity Sensing in Wild Vampire Bats. I chose this paper because I think it is super interesting to understand how sickness behavior can change pathogen transmission possibilities in a social network. So I'm going to explain to you very briefly what they did. So in the past, they have already done studies in the lab where they have shown that sick bats are more lethargic and they're less likely to interact with other bats, and they're less likely to call and to approach other individuals. And then this new field experiment wanted to recreate these experiments in a more natural setting. So what they did was to capture a wild colony of vampire bats in La Manai, Belize. They captured them at a site of Mayan ruins, and for the study subjects, they caught about 100 vampire bats, and then they divided the bats in two different samples, which was treatment bats and control bats. So they only kept the non-pregnant females, and then to create or to make the bats sick, they injected them with a solution of lipopolysaccharide, which is an immune-challenging substance that is part of the outer membrane of gram-negative bacteria. And what this substance does is to elicit an immune response on the bats, and they don't actually get sick, but they develop some of the sickness behavior or some of the sickness symptoms. And then for the other bats, which was the control group, they injected them with a saline solution. So after they did these two treatments, they also attached proximity sensors to continuously track the bat's social encounters over a time period in which the sickness effect of the lipopolysaccharide is supposed to be having an effect on the bats. So what they found was that just as in captivity, the sick bats were associated with fewer group mates, and they spent less time together, and they were less socially connected with other group mates that were very social and were very active. And another thing that they found is that, for instance, in the pairs of a sick bat and a control bat, they had on average a lower probability of associating and of spending time together. And for instance, in pairs of sick and sick bats, it was a lot less likely that they would have associations and spend time together. So this is very interesting because, it has received a lot of media attention, actually, this paper, because these bats are practicing sort of a social distancing, but it has been a little bit misunderstood because in the media it has been said that actually the bats are self-isolating themselves, but actually vampire bats don't do this. What they're doing is what I'm telling you is like a passive social distancing behavior, which occurs just simply because they are reacting to the sickness behavior. So this phenomena actually occurs in other bat species and in mice and probably in other mammals, but they are not self-isolating because they're showing altruistic behaviors. They're just spending less time together because they're slower and they're lethargic and, they have sick symptoms. So that's why they are interacting less with other bats. I think this is a really, really interesting experiment, also because these proximity sensors that they used are a really powerful tool to understand how changes in individual behavior can reshape how a social network is structured over time. And it has been, in the past they have been showing how social networks work, but there are a lot of limitations with the methods that were used in the past, because nest boxes of camera traps don't have the same reach as proximity sensors, because with proximity sensors they can actually continuously measure the times and the duration of the encounters among free-ranging animals. So this is something that is unique about this method. Well, of course, this part that I'm telling you is a very brief summary of the paper, and I really recommend you that you read it, and not only this paper but the past work that this group has done. I think it's super interesting and it's super inspiring, and well, I really think you should give it a check. And thank you for listening to me. I hope you enjoy the video, and see you next time.